exams and all. Yeah. Well, so this the purpose of this meeting is to interact with the mentor. Few of the mentors are already joined. So we'll start with bioinformatics project, then we will move to computational chemistry, then physics part 2. And after this meeting, okay, let me explain the process first. So today the mentors will explain what is the project, a brief about the project and how they plan to complete the project. And after this meeting, we will share the mentor's contact information. So you can definitely contact the mentor. You can discuss with the mentor. So before that, please go through the abstracts that we already uploaded in the website, and we already shared one PDF document too. So as I said, after this meeting, you have to contact the mentor before deciding things. And after 2 days, we will share the link to register and for the payment. So the thing is the entry to the project. So as you can see, there are multiple projects, and all the projects cannot accommodate all the people. So there will be restriction. Maybe 1 or 2 students can only accommodate in one project, isn't it? So that accommodation actually will be followed this first come first basis. So we will share the link. So if you join with one project, then it will automatically stop taking the other registration. So there are some number of students restriction too. So before that, this 2 days you can use to contact mentor and discuss and make sure that this is the project that you want to do. Okay. So yeah, that's the only thing we have to give. So now we can start this mentor meeting. First, I'm inviting Ankit Jana. He is one of the mentors in bioinformatics. Ankit is doing PhD in National University of Singapore in collaboration with Institute of Molecular and Cell Biology, ESTA, Singapore. And he has completed his master's in biotechnology from KIIT University, Bhubaneswar. And before joining his PhD roles, he worked as a research assistant at National Institute of Science, IIC Bangalore. And his research domain includes RNA, epigenetics, genomics, cancer, and stem cell biology. And there is another mentor, Sokriti. She also joined with Angit, and there are 2 projects in bioinformatics domain. And Sankriti, she is currently working as a lecturer in Midnapur College, West Bengal, and she completed her master's in statistics from Vishwabharati University. After that, she worked as research assistant in Ashoka University. And her research area is on biostatistics, image processing, and cancer data science. So these are the two mentors for bioinformatics project. So I welcome Ankit to brief about his project. So over to you.
Thank you for giving me the opportunity. I'll start by sharing my screen. Hope you all can see my screen.
Yes, we can.
So basically, there are a few projects on bioinformatics. I want to talk about those projects. So basically, I am Ankit. I have been introduced, so no need to introduce myself again. So I will talk about the project now. So basically, for this project, uh, basically there is no need of having prior knowledge of programming languages required. So if you are studying bachelor's or master student, if you have studied life science, then you can do this project. And in this project, what are the skills students can develop? So basically they will learn basic R programming, and also students can anticipate several significant outcomes that will enrich their understanding of genomic data science and RNA sequencing data analysis. Students will gain a deep comprehension of the intricate methodologies involved in analyzing bulk RNA-seq data, from quality control and differential gene expression analysis to pathway enrichment and cell deconvolution techniques. Students will also acquire practical proficiency in utilizing coding skills, particularly in
Uh,
programming languages. Hello.
Sorry to interrupt you. Actually, the slide is not moving.
Oh,
You are still at the slide. Yeah, now
uh,
it's. Can you make it what slide share mode?
now is it okay?
Now we can see the second slide.
Third slide, can you see? Is it changing
No,
now? It's not
no,
changing.
no.
Okay,
It's
wait a minute.
increasing to same.
I will stop sharing and I will
Yeah,
again
yeah.
start
That may be
sharing.
better.
Can you see my screen now?
Yes, we can see the screen.
Uh-huh. Okay, let me.
Yes.
Now
Now,
is it
now,
changing?
now. Exactly.
Okay.
Now it's working.
Okay. So what I was talking is, what are the skills students can develop by doing these projects? Basically, they will learn the basic R programming, and students can anticipate several significant outcomes that will enrich their understanding of genomic data science and bulk RNA-seq data analysis. Students will gain a deep comprehension of the intricate methodologies involved in analyzing bulk RNA-seq data. They will start from quality control, and then they will do differential gene expression analysis to pathway enrichment and cell deconvolution techniques. Students will also acquire practical proficiency in utilizing coding skills, particularly in programming languages such as R, to execute complex data analysis workflows. Additionally, they will develop their ability to write custom scripts and implement analytical pipelines to tailor to specific research questions, thereby enhancing their computational prowess in genomic data science. So basically, for this project, the tentative schedule is in week 1 and 2, we'll give you training on programming essentials. Basically, we'll give you some. We will introduce to the R programming. We will train you how to write codes. We will give you small tasks, and those tasks needs to be completed within certain period of time. Then we will track your your development on programming, like how you are learning programming or not. Then we will, based on your advancement, we will decide how to what should be the pace of our training. After that, we will for week 3, 4, and 5 onwards. We will train you on how to analyze bulk RNA-seq data. So basically, we will introduce to you for of various R packages and how they can be used for RNA-seq data analysis, and also we'll give you some assignments to complete. Then in week 6, we will basically work on ideation of project, like what will be your project, and what is the research question or biological question we are answering by doing this project. So we will try to have a theoretical background of that question, of that biological question, and we will be studying about that in week 6. From week 7 to week 10, we will work on respective projects. Like students will work on their respective projects, and obviously guidance will be provided in this period. Like you will not be alone. You will be given task, like you will be given the goal. You have to achieve that goal. You will you will whatever whatever programming you have learned so far, whatever training you got, you have to utilize just those skills to to apply to your respective project and get the outcome. So after that, uh, I will briefly talk about one of the project, like, which is the effect of interferon gamma treatment on melanoma cell lines. So basically, uh, this the raw gene count is available in GEO. GEO is a database where the genomic data are stored. So we will download those data, and we will clean the data. We will generate DEC2 object, and we will plot PCA plot, and we will look at the differentially expressed genes. We will make volcano plot and all, like, whatever in a transcriptomic or genomic paper, whatever data or figure you see, we will try to generate all those. So after that, we will, in project 2, there is transcriptome wide analysis of gene expression regulated by YB1 in glioblastoma multiforme. This is the title of that project. So basically, here also raw gene count will be downloaded from GEO database. Then we will do analysis, and we will try to answer our biological question, that why B1, is it how much is it necessary for for glioblastoma. So, and we will use various R packages to answer this question. So this is this figure I have given you to give an idea what exactly we are trying to do. So these images are taken from from different papers, different scientific papers. When you read different scientific papers, nowadays you'll get to see different transcriptomic data. So and it's very confusing for us, for people who are not from computational background, to understand this data, but it's very essential to know. So by doing this project, not only you will be understanding the how you not only you will be trained on programming languages, but also you will be trained how to read the data from the papers. So this is very useful. This will be very useful on your scientific career also. So now I will hand over to Sikriti for to talk about the other project.
Hello, everyone. Firstly, thank you for giving me this opportunity. I am Shikriti, currently employed as a lecturer at Medinburg College Autonomous. So let's start about the project. Um, so this project is basically on health informatics. In this project, there is no prior knowledge of programming is required. So I will try to introduce you to programming languages. You will able to improve. Sorry. You will able to improve your ability with skills including understanding of diabetes and related healthcare issues. And including risk factor symptoms and preventive measurements, which can be valuable in both personal and professional contexts. Student will gain hand-on experience in applying machine learning algorithms to real life worlds, understanding their strength and limitation through these real world problems, experiment with various techniques and continuous improvements of solution depend on outcomes. Studies will be improve their solving abilities, and also student will gain hand-on experience with using their coding ability to perform in complex data analysis, producing um, uh, in particularly with the programming language like R programming. Regarding this course, your tentative schedules are in week 1 and 2. You will train you on programming essentials. So you will basically introduce in R programming and also give you small task that you have to complete this with a certain period, and after that week 3, 4, and 5, you will train you in machine learning methods, and you will develop your skills which is required in this project. After that, week 6, concept of project and study related to the biological question, and after that, week 7 to week 10, student will work on their prospective projects, and guideline will be provided in this project. So the title of this project is predictive modeling for diabetes diagnosis, which is using machine learning algorithms. This dataset is originally from National Institute of Diabetes and Digestive and Kidney Disease. In this project, our main objective is, can we build a machine learning model to predict whether or not the patient is in the data set have diabetes or not, also determine which variable are most important in this predicting diabetes, also explore the relationship between variables, identify the patterns, and gaining insight into the dataset. So we will complete this task, this following tasks during this project. Thank you. If you have any doubts or questions, feel free to ask.
Yeah, thank you so much, Ajit sir and Sugidhi madam. So now, if you have any quick questions, you can ask. So as I said earlier, we will share the contact information of these mentors. So you can have a conversation. But even though, if you have any quick questions, participants side, you can ask. Yeah. Then I think as the participants can contact them personally. And one more thing we have to clarify here. Most of the projects are interdisciplinary, and the code is for our reference. And the code for the first project is BioRP01, and the second project is BioRP02. So that those details you can see in the website, and of course in the PDF that we shared. In fact, the second project is missing in the PDF. However, you can find it in the website. Yeah. If there is no questions, then we are moving to the second domain, that is computational chemistry. Yeah. So let me make sure that there is no question in this part, right? Yeah. Yeah, that's great. So let me move to the second, computational chemistry part. Let me invite Zubin sir to give a short intro on his project. Zubin Sivanarayanan, he is currently doing his PhD in ISAT and his master's in chemistry is from Victoria College, and he was a rank holder. and he cleared JRF and gate exam tries in chemistry and chemical science. So with this short intro, I'm welcoming Jibin sir. Jibin sir, over to you.
Thank you. Is my slide visible and I'm audible?
Yes, sir. Yes, sir.
So, hi everyone, good morning. So, today I'll be giving the project topics on the theoretical analysis on dyads. So, what is actually dyads? What are the purpose of dyads? So, that is what we are focusing at the beginning. So whatever we are doing in the laboratory in chemistry lab, it's a kind of mimicking from the natural phenomena. So here, the main purpose of making the dyads in the sense two moieties, it can be same or it can be different. That is what I represented here. It's hetero as well as homo. So hetero means we are connecting two different moieties by a covalent bond. Homo means we are connecting the same moiety by a covalent bond. So what will happen if this comes to by a covalent bond connections? So as we know that obviously if it is a heterodimer, it's A and B, right? So A and B both different have these have different electrochemical potential. So obviously one can undergo oxidation and other can undergo reduction in the presence of light. Right. So this is a phenomena will generate a charge separated state. So what is the need of this charge separated state? In natural phenomena, we have the photosynthesis phenomena, right? In plants. So in photosynthesis, there is a in photosystem 1 and photosystem 2 that we have already studied in our previous classes. So in photosystems, there is a component, there is also a kind of dyad system, which is a homodiad. There is a in natural phenomena in photosystem 1 has the means longest lived charge separated state is present in the photosystem 1. So whatever we are mimicking from the natural phenomena to the lab by doing with the molecules are not prolonged as the systems in the photosystem. Okay, still we are trying to get a long-lived or prolonged charge-separated state. So in heterodimer, obviously, if you are radiating light, so one of the component which has the easily oxidizable tendency will undergo excitation. From the excitation, it will donate its electron to the acceptor moiety. So there will be a charge separated state. How we can analyze the presence of the charge separated state? Okay, so the dyads is not only by means of covalently connected, but also means of the non-covalently. So by mistake, I have written covalently connected. Here you can see it's non-covalently connected. It's a kind of stacking. You can see that the first one. Here, the first one, it's a kind of slip stacked. So one, it's not exactly over to the other. It's a kind of sliply stacked kind of thing. So this will give us certain properties. And the second one that I'm pointing here, it's exactly one top over to the other, right? So this will give another set of properties. So this stacking arrangement is also kind of considering a dyad system. So if we are moving on to the next part, so this is how we are analyzing the presence of charge separated state. So this is only one system. Okay, so I'm not showing the how does exactly a donor acceptor system happening now. That anyway will come to a project. So here this moiety has homo and lumo part here, and this ruthenium complex has a homo lumo here. So in the same way, we will analyze a what you say. Suppose this compound A is connected by compound B. Okay, if this organic compound is connected to the ruthenium complex, obviously this ruthenium complex is a ligand, right? So that will give electrons, and this moiety will complex and this will accept electron. So obviously, if you are looking into the HOMO and LUMO, the electron cloud only focus on one of the moiety and the whole only localized on the another one. So that is how we will confirm where the charge separated comes, in which state the charge separated nature is coming, and which are the transitions. Transition in the sense, not only HOMO to LUMO, there are another transitions also possible. So there is a state which is just below to HOMO, that we can call it as HOMO -1. And there is a state which is just above to the LUMO, it is called it as LUMO +1. So that transition can take place from HOMO -1 to LUMO +1, or else HOMO -1, HOMO to LUMO +1. So there are so many transition can be possible. So which transition has this CT nature? So that can be visualized using natural transition orbital, how the transition behaves like. Okay, then the same thing we can use between how does the dipole moment vector, transition dipole moment, how does in both donor as well as in acceptor, how does it aligned? So from the transition dipole moment, how we can analyze the charge separated state. So these are all what we are doing in our project. So the basic tools for the calculation as well as the visualization of the cloud electron whole cloud, as well as the transition electron dipole moment, transition dipole moment, everything will be Gaussian software that will be using for the optimization, as well as the the UV visible absorption, vertical energy transition, multi-wave function we can use for this whole electron analysis, as well as the natural transition analysis. All the picture dynamics can be visualized using molecular dynamics, visual molecular dynamics, so which will give a manuscript quality images. And Theodore is also a kind of calculation where we can use for where does the hole is localized and where does the electron is localized. So this kind of calculations we can do using Theodore. So thank you.
Thank you so much, Jibil sir. Um, yeah. So as I said earlier, the same thing goes here too. So we'll share the contact information. So if you are interested with this project, you can definitely contact Jibil sir. And maybe if you have any, if you have any specific question now, you can ask maybe one or two questions. Well, as there is no, see, you can, I mean, question in the sense you can keep it in the chat box, or you can unmute yourself and you can ask. Both the ways are okay. So, if you don't have any question as of now, again, you can go through the abstract, and you can we'll share the contact information, so you can contact Jibil sir for maybe if you have any, if you need any clarity in this project. Thank you so much, Jibil sir.
Thank you.
So there are a couple of other two. Okay, let me share my screen first. Yeah. Hope you can see my screen. I'm Sabil. Currently, I'm an assistant professor, Department of Chemistry at VIT University. So here, in my under my mentorship, there are two projects. The first one is investigation on chemosensing mechanism with the project code ChemRP04. And here we are trying to explore the sensing mechanism of molecule, especially organic molecule, the receptor. Um, so the reference work you can see in these two papers that we already published. Um, so here we'll take a experimentally reported paper, and we will explore its mechanism in computational and theoretical perspective. So we have to do the DFT and TDFT investigation. So the sensing process we are doing for an anion recognition or anion ion sensing mechanism. So for that, we have to use Gaussian software, and we have to explore the proper mechanism. What are the transition states, the possible transition state, and we have to explore the interaction, as Vincent said, the MBO PES calculations to understand more details about this interaction, the non-covalent interactions. So this is the one project, and the second one is on a catalysis activity, asymmetric transfer hydrogenation of ketones. So those are in chemistry. I think I don't need to explain what is this hydrogenation. So here we are taking a reported experimentally reported work, and we are trying to explore the mechanism in computational and theoretical perspective. And the project code is Chem RP05. So here the reported one, this mechanism is already reported experimentally. So we are will do the computational calculation to understand what are the intermediates, what are the transition state, and how the energy things are changing, and how these interactions are happening in computational things. Again, DFT and DFT calculations we have been doing here, and we are going to use Gaussian software. So these are the two projects, and the proposed way to finish the project is the first week will be on literature review, and there will be a Gaussian training. So for the Gaussian training, we will give you there will be live thing. But apart from that, we will give you the recordings of previous workshop. So we already conducted a workshop on Gaussian software, and there is another workshop one upcoming workshop on Gaussian software. So you can watch the previous recordings, and you can understand, you can learn how you can use a Gaussian. Then we will fix the molecular systems that we have, and we'll perform the calculations. So the second month onwards, or after fourth week, we will more pitch into the major domains. So we have to do different calculations, the optimization, frequency calculation, the transition state calculation, IRC, again NBO, PES calculations. Then the last 2 weeks we are dedicating for writing part. You have to give a final presentation, and the final, the last week will be a final report submission. So each week, you participants have to give an update. What are the things you did? So this is how we plan to finish this project. Yeah. Again, so this is again the detail of this project. We will. You can find it in the website as well as in the PDF. Okay. If you have any questions now, you can ask. Or as I said earlier, we will share the definitely I'll share my contact information. So you can contact me, and if you have any if you need any clarity on this project, you can ask. And in computational chemistry, there are other two projects too, but the members are not available today, so we postponed their presentation in coming days. So again, you will get another mail from us for the same member meeting, and that is for that. Those, I mean, some of the members couldn't present here. So these are the computational chemistry projects. And now we are moving to physics part, computational and theoretical physics part. Let me invite Dr. Debashis Das. Sir, are you here?
Yes, I'm here.
Hi, yeah, hi. Yeah. So let me introduce Dr. Debashis. Currently, he is working as assistant professor in Department of Physics at Geetham Deemed University, Bangalore. And before that, he worked as research associate in the BIOS group at the University of Texas Austin, and another research associate. He also worked as a research associate in Martina Haven Group. I don't know whether I'm pronounce it correctly. In Roth University, Boston, Germany. And he did his postdoctoral research in St. Bose National Center for Basic Science, Kolkata, and his PhD in physics also from the St. Bose National Center for Basic Science, Kolkata. And his research domains include solvation science, that is intermolecular interactions in biomolecular processes with a combination of experimental and MD simulation techniques. And low temperature hydrogen bond dynamics in cryopretentant system. And hydrophobic hydration, low temperature hydrogen bond dynamics, microheterogeneity, and self-assembly of biomolecules. So these are the couple of research domains, or some of the research domains he is currently working on. So with this short intro, I'm welcoming Dr. Debashis Das to the platform. Over to you, sir.
Okay. Can you see my screen?
Yes, sir. Yes, sir.
Yeah. Thank you very much for this nice introduction. And thanks to the Spark Institute for giving me this opportunity to, considering me in their project. It's a very good initiative. So as he mentioned that I am working in a couple of other projects also. For this short-term project, I only interested in this understanding the intermolecular interactions and the role of hydration, actually the role of that intermolecular interaction, that is called the solvation science in biophysical phenomena. So but I am from physics, so my name is Debashis Dasmanta. I am currently working in the Department of Physics, Geetham University, Bangalore campus. So as I said that I am from physics, but it looks little bit of biology, little bit of chemistry, but it doesn't matter actually. So no need to any prior knowledge. So what we actually look for, we will put some biomolecules or biological relevant molecules. Let's say for this project, I will consider only small molecules, and I will put it into the water. So all the work will be in simulation. So all the work will be in GROMAT software. So people, if anybody already has a little bit of knowledge of GROMAT, it's very good. But if they don't have knowledge, it's okay. So we actually look for the solvation or the interaction between the water and that molecule. So because solvation is a very, very important phenomena. It may be this water molecule is very apparently look like an innocent molecule, but that plays a key role in biological or chemical processes. You can see in this figure that whenever you put some large biomolecule such as protein or
sorry to interrupt you here. Actually, the slide is not moving.
Visible?
It is visible, but yeah, now actually that we can now, yeah, you can share it again.
Okay. Window. Now it's visible.
Yes. Now it's in the solvation science.
Yes.
Second slide. Yeah.
Yeah. So yeah, as I said that whenever you put some large biomolecule such as protein or DNA into the water, so some water molecule around that biological molecule, which possesses very, very different phenomena compared to the bulk water. So we will compare this bulk water phenomena and that solvation water phenomena. That solvation water actually governs the whole characteristics or whole working methodology of that biomolecule. So in this project, we'll not consider the temperature dependent effect. We'll only consider the hydrophobicity and hydrophilicity. What is hydrophobicity? The part of that biomolecule which do not want to interact with the water molecule, and there are some part which wants to interact, preferentially wants to interact with the water molecule. And due to that preferences with water molecule and without water molecule, there are various phenomena is happening in the biological molecule. So let's say this one. So you can see the last figure. So this figure is the water clusters or the phase separation molecules inside that biological molecule. So here I have only shown the water-water phase separation. So there are also that biological molecule phase separations also. So all the work will be considered with this hydrogen bond definition, which is mainly the geometrical criteria between the two molecules. Regarding the project objectives, so as I said that I will mainly see the solvation behavior, how it goes around the hydrophobic molecule and hydrophilic molecules. There is a various changes. So we'll calculate different parameters to characterize this hydrophobic hydration and the hydrophilic hydration. I will mainly interested on the solvation configurations, solvation structures. We can, if time permits, we can also do a little bit of solvation dynamics also. But mainly solvation structure we will interested in this short time period. And we will ask this question that what is the connection between the solvation structure with the excess thermodynamic property? Because thermodynamics actually govern all this phase separation and the biologically relevant things. So we will ask this question that what are the connections between the solvation and the thermodynamics of the solution. As I said that we will do this project with the help of GOMAC software, but no prior node is needed. But because I am also not from chemistry background, so I also learned this GOMAC software. So students can think that we will together learn these things, and we will learn this how to perform the molecular dynamic simulations with writing some very short simulations code. So I usually write my own code, so that depends on what we want to look for. So this is not very hard task, I will say, but with these very small steps, you can have a very good knowledge of the science regarding your system. Regarding the timeline, I cannot really fix the timeline, but here I have written that month 1 will simulate the system, and month 2 will analyze the trajectories, because after the simulation, what we'll get the trajectories of those molecules at various time frame. And at month 3 we'll just compute the solvation structures. But during the whole period of time, we'll always focus on the scientific questions, like what are the role of those solvation in the thermodynamics or in the biologically relevant thing. Thank you all. If you have any question, feel free to reach.
Thank you so much, sir. Yeah, if anybody has any questions, you can keep it in the chat box. And I think we can move to the next vendor, Dr. Nawaz. Nawaz, sir, are you here?
Yes.
Yeah. So before going to that, again, let me confirm it. Do you have any questions on the project just now, Dr. Dabas was presented?
Sorry?
No. This is to participants. Yeah, okay. So now we can move to the next presentation, Dr. Nawaz. Dr. Nawaz MP, currently he is working as an assistant professor at MES KVM College, Kerala. And he is an alumni of IIT Delhi, and he did his PhD from IIT Delhi, and his post-graduation from NIT Calicut. And Dr. Nawaz has a rich research background. He worked as postdoctoral fellow at CERN Bangalore, and another postdoctoral research at ISER Trivandrum. So with this short introduction, I'm welcoming Dr. Nawaz to the platform. Over to you, sir.
Thank you, Dr. Sebil. And so I'll share my presentation first. Is it visible now?
Yes, yes, yes.
Okay, so I'm basically an experimental physicist. Most of my work is on experimental based. 90% of I work on experimental. But to support that one, we always look for this simulations, computational physics, so that we can actually get a better understanding of what we are doing as experimentalists. So in this project, I'll be basically assigning you with the work which will support the experimental evidences, or we can actually design whatever we need for these experimental things. So this is a kind of a project which is which can be a standalone project and which can actually support the experimental findings also. So there are a couple of areas I'm working on. One is plasmonics. You might have heard about plasmonics. Plasmonics is basically the collective oscillation of electrons. So these collective oscillation of electrons can actually give to a number of optical properties. So one of them is that localized surface plasmon. So before going to localized surface plasmon, what is surface plasmon? I will just explain. Surface plasmon is these collective oscillations. Actually, we can excite those collective oscillations using electromagnetic radiations. In bulk material, it is not possible to excite, but at the dielectric metal interface, it is possible to excite these collective oscillations. Those excitations are actually called surface plasmons. Now, if we restrict that surface of that particle, so basically, if you may go into nanoparticles, now we have very small localized surface, then this surface plasmon will become localized surface plasmons. So what is the importance of this surface plasmon is that these surface plasmon resonances actually depends upon the size of the particle, shape of the particle, as well as the composition of the metal nanoparticle. Suppose we have gold, suppose we have silver. These silver, gold, everything will give different surface plasmon properties, which depends upon the size of that particle, shape of the particle, and the composition of the particle. And very important thing is that it also depends upon the surrounding dielectric environment of the particle. So in this project, what we are looking forward is to first thing is the plasmonic response of the various nanoscale metallic structures. So we have different kind of metals, we have different shapes of metals. So we'll basically look into the plasmonic response of this different kind of metal. So it is a kind of a project where you can actually find out how these different kind of nanoparticles are actually responding to the shapes and the size of those particles. And the important thing about this plasmonic response is that it actually depends upon the surrounding dielectric environment also. So that property we can actually use for the refractive index sensing. So kind of it can be actually work as a sensor. So kind of suppose you have a sugar solution or you have a blood sugar. So if you want to find out the exact amount of blood sugar, that variation in the blood sugar can actually lead to the change in the dielectric environment. So looking at that change in the dielectric environment, we can actually back calculate what is the blood sugar or the amount of sugar in that solution. So in this project, we will be mainly focusing on how we can actually use these plasmonic properties for the sensing properties. So we will this project actually can contain 3 to 4 students. Each student can actually look at different material, different shapes, and we can compare this one. So once the outcome of this one will be, each project will be kind of at least will go to a conference paper, and then combine 3 or 4 students' work can combine to a journal paper. So that is how we have in the last project also, we have done the similar way only. The individual project actually sent to the conferences, and then the combined work we are actually planning to do in communicate in the journal paper. So that work is already the conference thing is already done. We have already students already have conference papers, and now that they are compiling it for the journal paper. So the next project is actually on the different aspect, which is the metamaterials. The metamaterials. Hello.
Sorry to interrupt you. Actually, the screen still we are seeing the plasmonics. Can you?
It's not visible.
It is visible, but it is not moving. So now metamaterials. I think
okay.
Even the last slide also, I think they didn't see. Can you just show? Yeah. Maybe you already explained it, but they didn't see.
So this is the outline of the project. So project is basically there are two kind of projects in the plasmonics. One is we are studying the plasmonic response of the different materials, and the other project is basically on the little bit of the application part, where we look at the sensing part of this materials. So this plasmonic response itself is a big project. We have to look at different kind of things, how these particles are responding to the plasmonics, and how this electric field is varying, how the hotspots are. So that first column is actually a complete project where we can actually have 3 to 4 students, and the other session is the refractive index sensing. So we are looking at the same plasmonic materials, and then on the application part, how we can use it as a sensor. That is the second project of this one. Now we move to the other portion where we have metamaterials. You might have heard about metamaterials. Metamaterials actually materials, artificial engineered materials, which will give properties which are not found in nature. So we'll give it will give such kind of properties which is not shown by the natural materials. So this is actually produced by the periodic arrangement of the nanostructures. So you can see the picture here. So this is one kind of such metamaterial. So we will simulate such kind of metamaterial. We will see how this metamaterial is actually behaving. So there are 3 projects in this metamaterial. One is, again, we'll use the same plasmonic material in the metamaterials. So now we have some plasmonic properties is there. Now we are artificially arranging it in the ordered manner. Then the plasmonic metamaterials will form. It will have a different kind of properties than the plasmonic materials. So that is one project. And the second project is that, okay, this is the project we have, plasmonic metamaterial, where we'll see the plasmonic response of a metamaterial. We'll also find out what is the electric field confinement in that, how we can use that for the molecular detection, and then we'll find out how near field and far field scattering is happening in these materials. So next set of materials is perfectly reflecting surfaces. So what is the importance of these perfectly reflecting materials? Is we don't have 100% reflecting materials. Okay, so we'll try to design a material which will give you 100% reflection in the visible region. Okay, you will find a lot of papers maybe in the microwave region, IR region, come 100% reflecting, but there are very few work which is already done in the visible region. So our aim of this project is to find out materials or combination of materials which will perfectly reflecting in the visible region. So this will again form a metasurfaces. Metasurface in the sense that will form the periodically arranged structures. In this case, we are using actually not metals. In fact we are using all dielectric materials. So like silicon dioxide, titanium oxide, those kind of material we'll use, and we'll try to get 100% reflecting materials. So the project is based on finding out the perfectly reflecting surfaces. So this is an example of such kind of material where we are getting 100% reflecting surfaces. So what is the importance of these 100% reflecting surfaces? We have n number of applications for this one. So those kind of things I'll explain you once you come to project. These are actually very important materials where in some applications we need actually 100% reflecting materials. The similar to this one, we have one more project where we are actually using this kind of perfectly reflecting material. But the property of this material is that it is not a huge high band of perfectly reflecting material. Rather, we have perfectly reflecting at a particular wavelength only. So here you can see that we have a perfect reflection only at violet. Here we have perfect reflection at blue. Like that, we have perfect reflection at different wavelengths. So these can be actually used as a reflecting displays. So our aim of this project is to find out which are the material or which are the combination of materials suitable for each wavelength. So we'll design a display in such a way that where each pixel in that display is actually our metamaterial or the meta surfaces. So this is so current system is actually all the displays are actually transmitting displays only, where we have a backlight and then the light is actually passing through that pixels. Here we have a reflecting display. So the important of the reflecting display is that we can actually use the sunlight as a source, and then the light, those lights will only get reflected, which we need that the picture, everything will be formed using that even with the sunlight. So we can actually reduce lot of power consumption by the mobiles, those kind of application which we can actually go for. This is one of the other project in the meta surfaces. We have one more project in the meta surfaces, which is again based on the perfectly reflecting idea, where we have IR shielding structures. So we'll find out such kind of meta surfaces in which it will transmit all the visible radiations and it will block all the NIR radiation. So if you look at the sunlight, almost more than 50% of the energy is actually coming from the IR radiations. So visible light is only 40 to 45% only. So normally the new houses, you can see that most of the designs are done using glasses only. So it will allow large number of light to come in. So we'll get more in the daytime, we'll get a natural lighting from that one. But the problem with the natural lighting is that it will also heat up our room. So if we can design a window in such a way that it will transmit only visible light and it will block all the near infrared radiations, so that we'll get such a kind of situation where we get a lot of light, but it is still cool. So that is the other meta surfaces we are looking for. So basically we have 4 different kind of projects. One is plasmonic materials, then plasmonic metamaterials, then we have perfectly reflection surfaces, then we have perfectly reflecting displays are there, and then we have IR shielding structures. So everything is in this one. We'll be doing all the simulation work using COMSOL multiphysics only. So this is you might have heard about this one. This is a kind of software where we can actually do many kind of things. So everything whatever I am explaining is only in the optical region we are looking at. So where we have an electromagnetic wave module. So we can actually do all these simulation using COMSOL multiphysics. We can design our meta surfaces. Then once we have ready with the meta surface, we can actually experimentally synthesize those kind of materials. So that part we may not be doing, but we can actually can lead up to what is the kind of design we need for these kind of applications. Okay, so that's all from my side.
Thank you so much, Dr. Nawaz. So again, if you have any question, you can ask. Otherwise, you can contact him. As he has a couple of projects with you, you can, if you are interested in any particular one, you can contact. Let me move to the next presentation. And that's by Dr. Aswil Ahmed. Sir, are you here? Aswil, sir?
Yes, Sabbil, yeah, I'm here.
Okay, thank you. Dr. Aswil, he completed his PhD and MPhil from Pondicherry University, and he did his postdoctoral research theoretical physics in Institute of theoretical physics at Institute of Mathematical Science, Chennai, India. He has worked on statistical mechanics of interacting self-avoiding work on 3-dimensional matrix. And currently he is working as assistant professor in I forgot the college. One college in Nagarjuna.
College.
Yeah, sorry for that. It's very
No,
yeah, with this short intro, let me welcome Dr. Aswin. Sir, over to you.
thank you, Sabil. And also, thank you. I would like to thank SIAS for organizing this wonderful program. And let me also uh, share my happiness in sharing this venue with other faculty members who are offering their projects. I was listening to many of them. They are all wonderful projects. And also, I would like to welcome the students who are present here. So, let me just try to switch. Is it changing?
Yes, it is.
Okay. So I'm not going into the details of the project. I will just give you what is an icing model. There are only 3 slides. So let us move to the second slide. Sorry, yeah, the second slide. What is an icing model? So maybe if you had a course on statistical mechanics, you might have already seen in statistical mechanics icing model. It is a model, a mathematical model used in statistical mechanics to understand the phase transitions in magnetic systems. But more than that, it is a theoretical framework, or usually we say that it is a toy model, using which you can study most of the critical phenomena and phase transitions in physics. And since you might already know what is the importance of critical phenomena and phase transition, since this is a toy model, understanding Ising model will and help you to understand many other phase transition problems in physics. And usually, most of the phase transition problems in physics will have a universality nature. So I will not get into the details now. So by studying Ising model, they can easily understand most of these things. So the second question is, how can we simulate an Ising model? So for simulating Ising model, usually I prefer basic programming languages like C, C++, or Fortran. If they have a pre-knowledge of any of these kind of basic level languages, then it will be very good. But if some students these days, most of the physics students, they are not learning these programs. Instead, they are either learning packages like MATLAB, Python, and Mathematica. And knowledge of these softwares are also means they can also do this same project using MATLAB, Python, and Mathematica. But the problem is that when we like to do hardcore programming, then these packages are not suitable. So there we will have to do something that we can see. So for similarity IC model, we will be using 2 algorithms. First one is the historically important, one of the earliest developed algorithms in the entire computational physics problems. It is known as Metropolis algorithm. We will learn the how to implement Metropolis algorithm using Ising model. But the Metropolis algorithm is a generally accepted algorithm, and it has a lot of application. Maybe in the next slide, I will just mention a few. The second one, which is a popular method and which is getting a lot of attention and a lot of publications in these recent days, is known as Wang-Landau algorithm. This is a recent algorithm which just came in 2001. But if you look at the literature associated with this algorithm, you can easily see that it is very powerful. A lot of variants of this algorithm are also proposed. And since this is a new, but these 2 algorithms, why did we select these 2 algorithms? These 2 are very easy to code. It does not take more than 10-15 lines to uh, code these algorithms. It is easy to understand, easy to code, and easy to implement kind of algorithms. But you can do a lot of physics using these algorithms. That is why we have chosen these 2 algorithms. Now, my last slide. What can we learn from this project? So, since this is a toy model in statistical mechanics, by understanding or by simulating Ising model, you will have a deep understanding in statistical mechanics. As I already explained, it is not limited to statistical mechanics. These days, these 2 algorithms, they are applied to condensed matter physics, quantum mechanics, high energy physics, and material science. And across cross-disciplinary application, it uh, yeah. In cross-disciplinary application, it is also used in business models then statistical models in biological applications. Many places it is applied. So what will a student gain from doing this project is that he will get algorithmic thinking and numerical problem solving techniques, and it will also help them to gain some programming proficiency. I think I can stop my presentation here, since no students have asked any questions so far. I am also not expecting any questions in my session also.
Yeah. Thank you so much.
Welcome.
So if you have any questions, you can keep it in the chat box. And we are moving to the the last presentation of today's session.
Sarabil, just one thing. Since I am
Sure.
little bit busy, I will be leaving now. If
Okay, it's okay.
students have
It's
any
okay.
other knowledge, or if there is anything, you can inform
Actually,
me later.
we actually will share the personal contact of all the mentors with the students. So
Okay.
they will contact the mentors.
Okay. If any student is contacting me, please write that you're contacting in WhatsApp or something. If you're contacting me, you please write that it is regarding to this project. Otherwise, sometimes I will not be able to reply immediately.
Yes.
Okay. Then thank you for
Thank you,
giving
sir.
this opportunity once again, and thank you all.
Thank you so
Bye.
much.
Bye.
So let me invite Dr. Sharaf Ali for his project presentation. Dr. Sharaf Ali, he is currently working as assistant professor at Salam Science College in Kerala, and he did his PhD from Pondicherry University in nonlinear fiber optics, and he published more than 10 international papers, and he supervised more than 30 MSc projects in his stay at the Salam College in Ladakh. So with this short intro, let me welcome Dr. Sharaf Ali. Sir, over to you.
Okay. Am I audible?
Yes, sir, you are.
Okay, thank you. Okay, I hope the screen is visible.
Yes, we can see the first slide. In fact,
Okay,
yeah.
now it is going, right?
Yeah.
Okay. Okay, thank you for inviting me for this presentation. In this section, we will be discussing about the photonic crystal fiber-based nonlinear applications, which include all my project topics. Myself, Dr. Sharaf Ali, assistant professor from Salam Science College, Erikode, Kerala. When you consider the optical fibers, which are very thin, flexible glass or plastic strand, along which the large quantities of information can be transmitted in the form of light pulses. And this slide, from this slide, you can see this is a brief historical.
Actually, sir, the slide is not so
Not
still we
visible?
are now still we are in the first slide.
Okay.
Yeah, now it's in second slide, but it's in the normal mode, not in the you may have to scroll down like that.
I think it's okay now.
Yeah, it's fine now. Yeah.
Okay, I will do like this. Okay. Okay, so this is just a history of this optical fibers. It all started from 1854 when John Tyndall demonstrated light bending of light through a curved stream of water. And from there, in 1991, we have discovered this photonic crystal fiber, which is a special class of optical fiber, which has a lot of advantages as compared to conventional optical fibers. And when you go to the photonic crystal fibers, these are, as I said, a special class of new class of optical fiber that obtain its waveguide properties from an arrangement of very tiny and closely spaced air holes, which go through the whole length of the fiber. And in this section, we will be just discussing the structure of photonic crystal fiber classification and applications, which are necessary for our project work. Photonic crystal fiber, when you check the photonic crystal fiber structure, you can see these kind of air holes in the three-dimensional figure. You can see all these are air holes which are running through the fiber length, and from this two-dimensional figure you can see these blue circles are representing the air holes, and this dark colored portion is the silica. So this silica will be having higher refractive index as compared to these air holes, and this region with the air holes will act as the cladding, and the center region will act as the core. So that is the basic principle of this uh, photonic crystal fiber, and that will be propagating light with the help of by the principle total internal reflection. Okay. And if you consider again the photonic crystal fiber, these are some of the manufactured photonic crystal fibers, which are these are the some images of that. As you can see that this is a solid core photonic crystal fiber, where if you'll be having the central core region which is solid, but here we have the hollow core photonic crystal fiber, where the center region will be hollow. Okay, so their mechanism of pulse propagation or light propagation will be different. So when you consider the classification of photonic crystal fiber, these are the two main classification. One is high index core fibers, where you will be having at the center, you will be having the solid core or the solid material. But in the case of photonic band gap fibers, you will be having void or the air hole at the center. So in this case, high index core, the light will be propagating by the principle of total internal reflection. And in this case, it will be propagating by using the photonic band gap effect. Okay, so these are the main classification of photonic crystal fiber, and there are many other classification of the photonic crystal fiber also. And now we'll go to the applications of photonic crystal fiber. There are a lot of applications. You can see the soliton pulse compression, supercontinuum generation, sensors, fiber lasers, etc. And we will be focusing on the first three of these applications: soliton pulse compression, supercontinuum generation, and sensors. And in this for this application, first we'll check what is actually the optical soliton. When you consider the optical soliton, these are some special pulses which cannot change their shape, or it will not be affected by any kind of disturbance while propagating through the fiber. So this kind of optical pulses are very important nowadays, because when you are communicating, suppose you are sending one pulse from here to some distant locations, for example USA or UK, through optical fibers, that optical pulse will be having some effect. For example, it will be affected by the dispersion, and the pulse will stretch. So these kind of effects will not be present in optical solitons. So these optical solitons are very useful for this kind of communication without having any troubles. So these optical solitons are further classified with the soliton order, which is given by this equation. As you can see from here, this first figure representing the first order soliton, so that will not change in its shape or anything while propagating through the fiber. You can see the distance. And when you consider n equal to 2, and this n equal to 2, which will depend all these parameters, we will discuss all those things in while if you are coming in for the project work. So when it is n is equal to 2, then there will be some periodic variation in its spectra as well as in time domain. Okay. So pulse width as well as spectral width will change periodically when you consider n equal to 2, n equal to 3, and so on. So these are the optical solitons. And what we are looking into this project for project work, we will be looking into the higher order soliton compression, or other kind of soliton compression also there, adiabatic soliton compression, self-similar pulse compression, all those things. So we'll be discussing only the higher order soliton compression. When you consider higher order soliton, when the n value that we discussed in the last slide, which is larger than 1, then you can have that higher order soliton. And when that propagates through the fiber, it is pulse width will get reduced after some distance. That you can see from here when you are considering second order soliton, which means n equal to 2. When n is equal to 2, then you will be having this pulse, input pulse, which is red color, as you can see from here, with large pulse width. That will be compressed, or its pulse will be shortened by pulse width will be reduced when it propagates to 2.28 meters of length of fiber. As you can see here also, for third order soliton, then the compression is much more higher, and you will get a very short pulse at the output of the fiber. Okay, so all this depends on what kind of fiber you are using, the structure of the fiber, the parameters of the fiber, etc. So by properly arranging the structure and parameters, you can decide what compression rate you need. In the last, also the last Nobel Prize also awarded for this attosecond pulse generation. Okay, so that is same procedure, but in that case we will be using normal optical pulse. Here we will be using solitone pulses. Okay, then we will move to the second application, that is second project work, which is the supercontinuum generation by photonic crystal fibers. Supercontinuum is nothing but a broad range of frequencies that can be generated from a single frequency. Okay, suppose if you are having a laser of red light, which is having some wavelength of 800, 700 nanometer. So then if you are sending that light through the optical fiber, and at the output of the optical fiber, you will be getting a broad spectra, which will cover the entire UV visible range to the IR range. That process is called as the supercontinuum generation. And this supercontinuum can be generated by three different nonlinear processes. One is solitone fission, second one is self-phase modulation, and third one is modulation instability. All these are different phenomena which will give you supercontinuum generation at the output of the optical fiber. And all this output or the supercontinuum will find applications in spectroscopic studies, biomedical imaging, broadband communication system, white light interferometer, etc. Okay, the third application is optical sensors. These are the optical devices which will sense one particular parameter from the environment and respond to some output on the other system. So this is if you are considering less just like a thermometer, where you will be measuring the temperature of these surroundings. Similarly, we will be having optical temperature sensors, where we will be using optical properties of the pulse which is propagating through the optical fiber to sense the temperature. So only that difference. Similarly, we can also sense different biomolecules, where we will be having biosensor, optical biosensors. Then you can use strain by using optical fibers. That is called as optical strain sensors. Then you have temperature sensors, etc. So all these kind of sensors are there, and we will be also developing some optical sensors based on photonic crystal fibers. That is the third work. And this is the last work that we have done to calculate the hemoglobin concentration in blood by using the half-cut photonic crystal fiber. So this geometry we have designed, then we measured what is the loss affected by the pulse while propagating through this kind of fibers when there is different kinds of blood coated on that surface with a different concentration. So analyzing the loss experienced by the pulse, we can say what is the concentration of the hemoglobin in that particular blood sample. So this is the last work that we have done. Now going to the what are the different steps that we'll do in our project. It is first one, we have to design the photonic crystal fiber, and we use COMSOL multiphysics software to design the photonic crystal fiber. We will fix all these parameters, geometries, all those things, materials, etc. And we will have some kind of geometry. You can use different kinds of geometry. You can use hexagonal geometry, you can use pentagon or square lattice, etc. Or also you can use different materials here. You can use a different other properties. You can vary like pitch, the distance between air holes, the diameter of air holes that you can be adjusted. You can fill liquids, etc. So all those things can be done in COMSOL, and we'll finally get the effective refractive index of the light which is propagating through the optical fiber, and also effective area. And by using that, we will be calculating dispersion and nonlinearity. And for dispersion also, we need this equation to be solved numerically. We use MATLAB for that. And then we have this nonlinearity here. This you need A effective from the COMSOL to calculate the nonlinearity. Also, you can calculate what is the loss in the fiber by using this equation. And with respect to the application, which applications we are focusing, we can calculate either dispersion, nonlinearity, or loss, confinement loss. in photonic crystal fiber. And all these things are very much dependent on fiber geometry. As you can see from this figure, the dispersion can be shifted very largely with a very small change in d by lambda ratio, which is the diameter of air hole to distance between the air hole ratio. So only by changing the geometry, you can change the dispersion properties of the fiber, the nonlinearity properties of the fiber loss also you can change. So this is a very easy method, or which will give you more vulnerability in the case of attaining an optimized structure for the fiber as compared to the step index fiber. In step index fiber, we cannot change these properties in this way, because you need to dope different material into that. All those things are there. So there are a lot of advantages when you consider the photonic crystal fiber. Apart from this step index fiber, conventional step index fiber. So these are the softwares that we will use in the project. Mostly we will be using 2 of them or 3 of them. We will not be using all of them. But these are the softwares that we will be using. And for more details, you can visit my Google Scholar ID, where you can find different topics or papers you can get. And also regarding the publication, we have published in all the last years, 3 years, we have published the students' project work in international journals. So it only that the deciding factor is how the students work with respect to that topic. If the students work well and they are having that passion towards the topic, then that result will be good, and that will be published in international journals, or at least in conference. So that's all about the project details. Okay, thank you for listening.
Thank you so much, sir. So now we came to end of today's session, and as sir rightly mentioned, it more than which project you are working, it's very important how you are approaching to the project, how serious you are to complete this project. That is very important. And there are a couple of other members that couldn't attend today's meeting. One, Dr. Bibab, he is currently working as assistant professor in Department of Chemistry at VIT University. Another is Dr. Sumit. He also working in Department of Chemistry at VIT Bhopal University. So both both these projects are more aligned to biochemistry, and they are using domains to explain the interactions. So that we will plan in coming days. And yeah, and another one, Dr. Shafiq Ali, he's in the physics background, and he also couldn't attend today's meeting. So those mentor meeting we will arrange in coming days. Yeah, so we will give the recordings. You can watch the recording. Some of the participants said they have exam tomorrow and all, and they couldn't attend today's meeting. Yeah, so if you can watch the recordings again, and you please go through the abstracts that we already uploaded in the Google, I mean in the site. And we will share the contact information of all the mentors. You can contact them. It's good if you contact them personally and ask your doubt. Yeah, and after today's, we'll share the registration link, and that moment you have to fix which project you want to do. When filling the registration, you have to tick which project you want to do. Yeah, so that's about today's meeting. Thank you all the mentors joining today's meeting and give a wonderful presentation of your project. And thank you participants for listening these things. And if you have any questions, you can ask. Otherwise, we will wind up the session.